All right. This video is about the online lab. I hope everyone has watched uh, yes. I hope everyone has watched the Watch Before Completing Genetic Technology Lab. And now I'm going to do the Genetic Technology Lab. So you're going to click on the PDF. You're going to find this and go to this place and perform a virtual PCR. Let's go. I think I have it set so that you can hear the computer audio, but if not, well, I mean, sorry. All right. The human genome is made up of 3 billion chemical base pairs. My goodness. Scientists often need to isolate a very specific segment of DNA from within a vast amount of genetic material. Since this segment is just one tiny piece of the genome, they need many copies to have enough to work with. Yeah, so basically you're starting out with a tiny piece of DNA, like at a crime scene or something, you like scrape some blood off of the ground, and then you've got to make more DNA so that you have enough to work with and do tests with. Cell nucleus. The man who invented PCR, I think his name is Perry Mullins. Perry somebody. He won the Nobel Prize. PCR basically revolutionized science. The polymerase chain reaction, or PCR, can generate 100 billion identical copies of a specific DNA sequence in a matter of hours. Yes. As the inventor of PCR explained, the technique requires no more than a test tube and a few simple reagents. Reagents are things that you put into the reaction, and a source of heat. Look at all that DNA. Ooh, here's some hair. To perform a PCR reaction, you need DNA, yes, that has been extracted from cells. Because the purpose of PCR is to make more DNA copies, you don't need very much DNA to start the reaction. For example, you could extract DNA from a small sample of blood, skin, saliva, or even hair follicles, something you might find at a crime scene. Start by moving your extracted DNA into a special PCR tube. PCR works by heating and cooling the solution over and over, so PCR tubes are designed for even heat distribution. All right. Okay. Drag the extracted DNA. Our tube. There we go. Next, add primer 1 to the PCR tube. Primers attach to sites on the DNA strands that are at either end of the segment you want to copy. They are powerful tools for copying very specific DNA sequences, since there is almost no chance that they will target the wrong sites. It's true. All right, primer 1, here we go. Hope you can hear the sound effects, but if not, they're kind of easy, like. Now add primer 2, which will attach to the second site. Okay. Come on, primer. Release into the PCR tube. Okay. Add nucleotides to your PCR tube. Nucleotides are the A's, C's, G's, and T's that make up the genetic code. These genetic building blocks will be used to create billions of DNA copies. All right. So there's nucleotides in this fluid. Put it in there. Last, you'll add DNA polymerase to the PCR tube. DNA polymerase molecules act like tiny machines that read the DNA code and then attach matching nucleotides to create DNA copies. This particular DNA polymerase has been specially selected to withstand the high heat of the PCR reaction. Yes, it's called Taq polymerase, TAQ, thermo aquatic, maybe? Something thermo. Right, now we got the enzyme in there. Yeah. Now that the PCR tube is filled with all the reaction components, you'll place it into a DNA thermocycler. The machine can precisely heat and cool your tube at specific times during the next hour. These changes in temperature are crucial for making the reaction work. Yeah, it basically heats it and cools it, cycling over and over. Okay. before these machines were invented, people had to have like a vat of ice and a vat of hot water and just stand there and switch the tubes back and forth for an hour. Right, go, start. Oh yeah. See what's happening in here. DNA, cycle number 1. There's a lot of cycles, y'all. Target sequence, that's what we want to amplify, meaning to get more and more of. Inside your PCR tube, cycle 1 has begun. The thermal cycler heats up to 95 degrees Celsius. That's 203 degrees Fahrenheit, good lord, which is almost boiling. At this temperature, the DNA double helix separates, creating 2 single-stranded DNA molecules. Aha, it does because it's so hot, it melts the hydrogen bonds. And you cool it down so that hydrogen bonds can form. Now the thermal cycler cools down to 50 degrees Celsius, about 122 degrees Fahrenheit. At this temperature, single-stranded DNA molecules naturally attempt to pair up. However, there are many more primer sequences than DNA strands in the tube. The primers crowd their way in and lock onto their target before strands can rejoin. Oh yeah, look, we're doing DNA replication here, DNA polymerase. The thermal cycler now changes the temperature to 72 degrees Celsius, which activates a DNA polymerase. When DNA polymerase locates a primer attached to a single DNA strand, it begins to add complementary nucleotides onto the strand. It continues until it gets to the end of the strand and falls off. Cycle 1 is complete. Click the next button to begin cycle 2. Okay, there we go. Same 3 steps happen in cycle 2. The temperature is raised again to separate the DNA strands. This is called melting the DNA. So the DNA is melted, meaning the heat is raised so that the hydrogen bonds between the 2 DNA strands cannot form. So they separate. Then we cool it down, we add the primers. The temperature is lowered so the primers will attach. Add the DNA polymerase, and there we go. And the temperature is raised again slightly to stimulate DNA polymerase to copy the strand. Click the next button to begin cycle 3. There we go, over and over and over. Oh, there we go, that's what we want. During cycle 3, your desired products begin to appear. 2 strands that begin with primer 1 and end with primer 2. These are DNA copies of just the segment of DNA you have targeted. Although there are only 2 desired fragments at this stage, as you continue with the cycles, these products will quickly become the majority. True, over and over and over. At the end of cycle 4, you have 8 fragments that contain only your target sequence. Now you have 22 fragments that contain only your target sequence and only 10 longer length copies. Where are we right now? Cycle 5. Click the fast forward button to run more cycles. Good lord. After 30 cycles, there are over 1 billion fragments that contain only your target sequence and only 60 copies of the longer length molecules. You now have a solution of nearly pure target sequence. Cool. Well, I guess that's all. Start over, back, yeah, none of that. All right, there you go.